My name is Olga Tarkhanova, and I'm very happy to host today's webinar, especially after quite a long break. And I'm very happy that at this webinar that we start our new series of webinars with such a great topic as DNA-encoded libraries. Before I introduce our guest speaker, I would like to remind you like a few facts about Chemspace. Yeah. Different type of search on our platform, like similarity search, substructure search, and of course exact match. Our IT team provides really great solutions and launch different custom integrations with ERP systems to simplify purchasing process for companies. We also do compound management, and quite recently we launched a new panel of services, hit discovery services. And today I will stop a little bit on these services. There are two main approaches we offer to our customers. The first one is called vSynthesis approach. And this is completely virtual approach, which allows to perform gigascale docking really huge spaces like in real, more than 60 billion molecules. To perform this approach, we need to have a crystal structure or NMR and cryo-EM structure. And yeah, having this, we will be able to perform a search and discover some hit molecules. Another approach, crystal structure first approach. It's a great combination of experimental approach and virtual approach. It starts with fragment screen using a crystallography technique, and based on the fragments which will be identified as after fragment screen, custom space around these fragments will be designed and docked using different approaches. And the last one, it's a DEL approach. It's a great approach which starts with DNA-encoded library screening, and then using the results of this DNA-encoded library screening, ML model will be built and applied to accessible spaces. Yeah, a few words about vSynthesis. So this approach does not require full enumeration of the huge spaces, but at the same time, a docking accuracy is comparable with a classical virtual ligand screening. This approach allowed to get hit rates and perform screening quite fast. More than that, it allows to work with like real huge spaces. Like for now, we work with 60 billion molecules. Crystal first approach, as I mentioned, it's a great combination of experimental and virtual approaches. So what we need to perform this approach, we need to have like a crystal structure of a few fragments, and then based on these fragments, we will build chemical space around these fragments using in real, dock these molecules and identify hit molecules. And of course, DEL approach. A DEL approach starts with DEL screening, and as you all know, DEL screening can generate like a huge amount of data, and this data can be used to build ML models, which can be applied to accessible spaces. It could be in stock set of compounds. It could be in real. So it's a really great approach, and of course everything starts with DNA encoded libraries, and we are happy to have a partnership with HitGen company. We have been working together like for many years already, and I'm really proud to introduce you Vice President at HitGen, Dr. Guansai knew him. Yeah, he got his PhD in organic chemistry in Shanghai Institute of Organic Chemistry. Then he did a postdoc in Georgia State University. And in 2016, he joined HitGen company as associate director of chemistry. And he went like all the way up to vice president, and since president and head of discovery chemistry unit at HitGen. Okay, Guansai, the stage is yours. I will change the presenter. Okay, yeah. Give me a sec.
Okay, thank you, Olga. So can you see my screen right now?
Yeah, not yet.
Hold on a minute.
Give me a sec. Okay.
You can see it, right?
Yeah. No. Give me a sec. I will change. Yeah. Now. Okay. Okay.
Yeah,
try to.
It should be working.
Yeah. Now I can see.
Okay. Yeah, thank you, Olga. So to make sure my network working well, so I just turn on my video from my side. Okay, to get the full screen.
Yes.
Okay. Yeah. So thank you so much, Olga, for the kind introduction, and especially thank Chemspace to give me this opportunity to present our work. So good morning, good afternoon, and good evening to the audience around the world. I appreciate the opportunity to be with you today. So the subject of my presentation today is OpenDEL as a self-serve DNA-encoded library kit benefit your drug discovery research. So my presentation today is divided into 5 main sections. First, let's begin with a brief introduction on the concept of DNA-encoded library technology. So DNA-encoded chemical library is often abbreviated as DEL. In 1992, Professor Brenner and Lerner first proposed the concept of encoding a chemical library with nucleotide tags. At the time of their disclosure, they presciently recognized significant challenges. First, DNA-compatible chemistry tools are quite limited. Amide coupling was the main chemical reaction used for DEL synthesis. So the DEL molecules in early years were quite peptide-like. Second, in 1990s and in early 2000s, the next generation sequencing technology that are used for identifying ligands with high affinity for a biological target was immature, and the NGS cost was very high. Although the groundwork of DEL was laid in 1990s, the field set domain until the mid-2000s. Thanks to advance in DNA-compatible reactions, selection methodologies, next-generation sequencing and DNA analysis, DEL technology allows the construction and screening of libraries of unprecedented size, which has led to the discovery of highly potent ligands, some of which have progressed into clinical trials. This slide showed a basic construct of DNA-encoded library compounds. Millions or billions of DEL compounds with the same generic structures constitute a library. A DNA-encoded library compound structurally consists of 3 parts. So this is a typical 3-cycle library with 3 chemical variants on small molecule parts. On the right part in the slides is a small molecule moiety. On the left part is a DNA species to encode the small molecule identity. These 2 components are covalently connected together with a long flexible linker. So PEG linker is in most cases. So let's suppose a small molecule on the right side constructed from 3 moieties shown in different color here. When the building block 1 shown in red is chemically attached to the linker, the DNA codon is enzymatically ligated accordingly. The same approach applied to the assembly of building block 2 and building block 3, and DNA codons 2 and DNA codon 3. So the synthesis of DNA-encoded chemical library compounds on the one hand is to construct those small molecule structures. On the other hand is to build the relationship between DNA tags and small molecules. So unlike traditional high throughput screening, the DNA-encoded library compounds typically pulled from multiple libraries are incubated with a target that is immobilized on base. So strong binders are attracted by the target and bind to them. Those that do not bind to the proteins are washed away by shifting elution conditions. So strong binders and isolated and collected. To increase the sound to noise ratio, the collected samples are incubated with fresh proteins again. So normally after 2 to 3 rounds of selections, the samples are collected for PCR amplification. So thanks to the next generation sequencing technology, the DNA barcodes are read out and the chemical structures are decoded and accessed by reading DNA tags. In contrast to traditional technology, DEL no longer requires a separate experiment for each compound. Instead, the experiments are performed in a single tube. DEL has reinvented the drug discovery process and is able to deliver screening results in 6 to 8 weeks. DEL has been proven to be a powerful tool for integrating biological targets by accessing vast chemical space. Beyond that, DEL screening has many other advantages over traditional approach. First, easily access trillions of physically existing compounds. Second, saving important target consumptions. Third, more cost-effective. DNA-encoded chemical libraries have been widely explored and applied for the target selections. A number of novel small molecule ligands have been generated from DEL selection toward a diverse set of therapeutic targets. Some of them have been progressed into clinical stages. Encoded library screens uncover several different protein-protein interaction inhibitors and compounds binding to the surface of GPCRs, kinases, phosphatases, transcription factors, etc. The compounds identified from DELs are pretty diverse, including traditional small molecules and microcycles. Next, I will show you some application cases at HitGen. So this is a collaboration project with Pfizer. NAA50 enzyme is a member of the N-alpha terminal acetyltransferase NAT protein family. Compound 1 is a no-potent NAA50 inhibitor, which doesn't look drug-like. The DEL selection is to identify potent and selective NAA50 inhibitors with improved physical chemical properties relative to compound 1. During the study of NAA50, we realized that the NAA50 protein involved conformational change in the catalytic process. So besides the selection against able NAA50, we set up 3 additional samples, including addition of cofactor acetyl-CoA, CoA, and black control without proteins. So this is one of the advantages of their selections compared with traditional high-throughput screening by setting up multiple parallel selection experiments. So finally, we identify drug-like hits with single-digit nanomole potency. So GPCR belongs to a superfamily of cell surface singly proteins that have a pivotal role in many psychological functions and in multiple disease, including development of cancer and cancer metastasis. Current drugs that target GPCRs, many of which have excellent therapeutic benefits, are directed toward only a few GPCR members. So therefore, huge efforts are currently underway to develop new GPCR-based drugs. So this is a case for screening of a solubilized and stabilized GPCR target using our DEL technology. Reported compounds were cyclid pentapeptide with molecular weights around 900 Dalton. DEL campaign derived small molecule with similar potency with drug-like properties. Protein-protein interactions represent a vast class of therapeutic targets both inside and outside the cell. The human endocolon has been estimated to cover 400,000 protein-protein interactions. L17A is an interleukin cytokine whose deregulation is implicated in autoimmune disorders such as psoriasis, and antibody against the L17A pathway are now well established and very effective treatment. So low micromole small molecule identified using our DEL selections. So this project is our internal project. The project is now progressed into PCC stages. Transcription factors are DNA binding proteins that play a key role in gene transfection. Transcription factors have traditionally been considered undruggable by small molecule ligands due to significant structural disorder and lack of defined small molecule binding pocket. So after our DEL screening for this target, 30 compounds directly identified from DEL screening were confirmed with nanomole S50s. The most active compound has S50 of 15 nanomole. With the discovery of PROTAC 20 years ago, targeted protein degradation has changed the landscape of drug development. To expand the reach of TBD to degrade other hard-to-drug proteins, novel ligands for the target need to be identified. This is a protein-protein interaction target. So after their screening campaign and optimization, a series of nanomole small molecules were finally identified. Then we conducted optimization work to design, make, and evaluate bifunctional molecules. This project is currently at IND in enabling stages. DEL technology is also powerful to find the ligands of E3 ligases. Thalidomide and its analog as CRBN binders are majorly used for PROTAC development. With DEL screening, we successfully discovered novel CRBN binders. Compared with thalidomide analogs, the new CRBN binders are quite structurally different. The new CRBN binders are evaluated by replacing thalidomide for BRD4 and BTK degradation. The new PROTAC molecules gave better DC50. The new CRBN binders are being further evaluated for various POI degradation in vitro and in vivo. The value of DEL technology is far more than identifying DEL hits only. DEL is constructed by combinatorial chemistries. In principle, a series of compounds called compound clusters are enriched in the selections. Therefore, the post-selection data is very important for medicinal chemists to analyze structure activity relationship and structure selectivity relationship. And for downstream hit optimization and lead optimization. For this case, you can clearly see three elements that demonstrate crucial for structure activity relationship. First, for this key scaffold, triazo substructure demonstrates the best compared with oxidazole, isoxazole, and oxidazole. On the left side of the hydrocycles, both aromatic ring and aliphatic ring are tolerable. And also the linkage between the hydrocycles and the scaffold also is important. In this case, CH2 is preferred. So finally, a 98 nanomole hit was quickly identified after post-selection data analysis. As Olga just mentioned that the post-selection data is also quite useful for prediction of active compounds outside of their compound chemical spaces. After their selection, we identify three series of compounds with activity ranging from 1 micromolar to 4 nanomolar. These DEL compounds share the same cyanide moiety. We trained models using DEL selection data and identified active molecules from a large commercial collections. Finally, the 2 series of compounds are finally identified. The first series of compounds contains the same moiety as DEL molecules. The heat conformation rate of this series using DEL machine learning is very high. 83% of the compounds were tested as active. The second series is quite novel and is structurally different from DEL hits. 24% of selected compounds are proved to be active. The highest activity is 19 nanomolar. As what we mentioned before, DEL selection data is very useful for medicinal chemists and computational scientists. In conclusion, DEL is one of the most powerful heat generation sources for early drug discovery. Although many companies and institutes have already developed DEL platform, still few organizations possess the DEL platform. The synthesis of DNA-encoded chemical library is an enormous investment on chemical building blocks and DNA tags. And development of DELs requires multidisciplinary expertise, like DEL chemistry development, DEL synthesis, DEL selection, next generation sequencing, and data analysis. It could be very helpful for researchers to have DELs commercially available for direct use. What's the idea on shelf DEL products? What are features of these DELs? For users, some elements may be considered before ordering DEL products from vendors. The product should be readily available. The molecule in the libraries should be drug-like and diverse. Users is able to access all DEL information, including chemical information and coding information. Some of users are not very familiar with the use of the DELs, and they may hope the guidance from vendors for DEL selections, sequencing, and data analysis. So finally, the product is affordable. For all these reasons, OpenDEL will be your choice. So this is what we will mention the OpenDEL. So currently, this OpenDEL was first launched in 2015. The kit includes 30 DELs 2 billion compounds, and also it includes 10 each kit includes 10 selection samples that are sufficient for 2 to 3 target selections. And this is a self-service DEL product, and the users can obtain these DEL kits in-house to enable our user to explore DEL campaign without sharing target information. And also, importantly, we will disclose all of the DEL information, including the building block information and codon information to our users. So the customers are able to analyze the structure and activity relationship trend, or run AI machine learning using building block and codon information. And also, because as I just mentioned that all of the building block information has already disclosed to our customers. So there's no structured disclosure fee, no license fee for hits discovered from OpenDEL. And the only thing the customers will prepare is the target materials. So especially for the medicinal chemist, when they obtain the OpenDEL kits, they usually care about what kind of the molecules in the OpenDEL kits. So there are some of the elements they may consider. One is the library design. The second one is molecule diversity, and the last one is molecule drug likeness. So let's first talk about library design. So we will have 2 examples to show you how we design our library into the OpenDEL kits. So here is just one of the examples. So we know that in 2014, there's a GA Medical paper published a review paper to talk about the indole-focused compounds. So at that time, there are 17 different drugs approved by FDA. And it's very interesting that there are 15 different drugs contain 3-position modification on indole ring. And also there are 12 different drugs contain modifications on the 5-position of indole ring. These are just some of the examples of the drugs. So then with this one, so we just want to cover the chemical space of indole focus library. So here it's just some of the example we have already put into the OpenDEL case. This is a 3-position modification focused indole library. So before that, so we haven't had those kind of the chemistry available for the library construction. Then we developed this kind of the chemistry and published in 2019 on organic laters. And we use a 2-step chemical conversion. First step is using the aldehyde to do the addition on 3-position of indole, and then to the reduction to the dehydrylation to get the indole ring modification on 3-position of the indole ring. So the reason we put the aldehyde here, because in the commercial source, so there are thousands of different aldehydes available. So that will increase the diversity of the molecules on the 3-position of the indoles. The second example is also the indolazone focus library. So here is just some of the FDA approved drugs and also some of the active compounds reported in literatures. So all of them contains the indolazone core structures. So at that time, we also consider whether we can include those molecule, those core structures into our OpenDEL collections. So before that, there are different kind of the chemistry reported to construct these kind of the structures, including using the metal catalysis reactions, using the high temperature reactions. But unfortunately, very few conditions can be compatible with DNA species. So then we developed a DNA compatible chemistry just in using the this dibro acid as a reductive reductant to reduce this kind of the natural group and then to the reclosure reactions. So with this one, so we just do the substrate scope study, and also this kind of the also published on Organic Later in 2020. So so you can see that those kind of the reaction quite diverse. Either the aromatic ring and aliphatic ring can be tolerable to get this core structure constructed into the libraries. So this library is also in the OpenDEL collections. So the second one is that to consider the molecule diversity. So one of the elements we have to consider that to increase the diversity of the molecules, so we have to make sure there is enough diversity of the building blocks used in the OpenDEL. So currently for the OpenDEL collections, we have already used more than 20,000 different building blocks for the library construction. Though they contain the different types of the building blocks, including the amines, carboxylic acid, aldehyde, boronic acid, diamines, etc. And we can see that for the amines, we use more than 7,000 different building blocks, 6,000 different carboxylic acid, and also not only in one chemical reaction using for example, for amines, the different kind of the chemical reaction used for amine into the library construction, including the acylation, SNR reaction, reductive amination, Foucault coupling, and also the benzoimidazole ring closure, and also click chemistry. So the other thing we consider is that topology diversity of the DEL molecules. Here, if we on the left side of this slide, we just show the topology distribution of the 2 billion compounds of the OpenDEL, and also the different library design shows very different topology distribution For example, for this one is a building block combination library. It looks like it's just the linear library. So the topology is a much more arranged to this kind of the zoom. And the second one is a scaffold-based library. So using the SP3 rich scaffold. So this the topology distribution is quite reasonable. And the last one is the hydrocycle focus library. So this is a much more planary topology. So that shows the quite diverse topology diversity of the molecules of OpenDEL molecules. So last one is that we call it diversity. Is that so? Although we just mentioned that there are 30 different independent DELs, but so we in this OpenDEL collection we have more than 30 generic structures. Currently, in total, we have 56 generic structures into the OpenDEL collections. We take one of the OpenDEL as an example. So you can see that in the cycle 1 of the library synthesis, we use the different types of the building blocks, including the amino acids, aldehydes, diamines, and primary amines. So after that, so we pull them together to do the cycle 2 acylation and cycle 3 reductive amination. So in that case, although this is the only one library, but it contains very diverse molecules, you can see that for these amino acids and diamines, it looks like the linear molecules, right? And for the aldehydes and primary amines, that is much more like the branched molecules. And also you can see that even we use this kind of the protocol for the library constructions, still we can see the different outcomes for the library screening. For this target, we call it target X selections. So after the selection, we can clearly see only 2 types of the building blocks can be enriched after selection, including the aldehydes and the primary amines. But there's no enrichment from the primary amino acids and diamines. So that demonstrates that probably the topology of the molecules could be very important for this target. And the last one is that what we call it, molecule diversity, because the medicinal chemistry is much more care about whether the molecules in the libraries are drug-like. So we just show the molecular weight distribution and A log P distribution of all of the 2 billion compounds. So you can see that for the molecular weight, so 90% of the encoded molecules gave molecular weight less than 550. And 91% of encoded molecules give A log P less than 5. And most importantly, for these OpenDEL collections, there are no 4 cycle DELs. So there's only 2 cycle DELs here to make our DEL molecules as druggable as possible. So after introduction of our OpenDEL library design and the other things, next, we will talk about the workflow of OpenDEL as a product. So first, the customer may purchase the OpenDEL package from us, and then we will send the OpenDEL kit to our customers. So along with the OpenDEL products, we also provide the information of the OpenDEL, including the building block information, codon information, and also the OpenDEL selection menus, to make sure our OpenDEL customers can use those products for their own target selections. So after the selections, so so this is just the option from our customers. The customers can choose to do the next PCR and next generations sequencing by themselves, or send the sample to HitGen for we do the next generation sequencing for the customers. This is the choice from the customers. So after that, so we can help the customer to do the data analysis and the hit proposal. So then the customers will involve into the data analysis stage to do the hit proposal and do the off-DNA synthesis and the protein activity validation. So this is just the whole OpenDEL workflow. And also during this process, so we are very appreciate if some of the customer gives some of the feedback, either positive feedbacks or negative feedbacks. So here is just one of our customer is a US biotech company. It's called September. They are focused on GPCR target study. So they give us very good positive feedback on the OpenDEL, and they also use this OpenDEL product successfully identified functional validated hits for multiple GPCR target. And also, the most importantly, we for our OpenDEL, they feel this OpenDEL product we are very open and also very flexible for the collaboration. So here, so if you are interested in our OpenDEL, so just shoot us the email, either from the BD email. Also, if you have some want to some of the technical discussion with us, and also you can send the email to the OpenDEL@HitGen.com. Also, shoot the email to myself directly. Okay, so the last one is that so we will give you a very quick introduction of our company. So our company called HitGen. HitGen was founded in 2012, and it's so constant. So today is our 11th anniversary for the HitGen company. So the company is headquartered in Chengdu, China, and also we have 2 subsidiaries outside China, including Vial in UK, Cambridge, and HitGen Pharmaceuticals in the US. And there are more than 500 employees, and in Chengdu we have 20,000 laboratory and office spaces. So in 2020, so the company is listed on Shanghai Stock Exchange. So for us, so we are currently focusing on innovative drug discovery research platform. So we have already built our technical research platform from target to IND enabling stage. So except the GLP safety, we have already set up all of the facility for our own project and also for services. So our key technology is to discover new molecules for the target. So currently we have 4 key research technology platform. One is DNA-Encoded chemical libraries. Now we have 1.2 trillion encoded molecules, and also in the past years we have already screened 51 different target classes, and almost 80% of the functional success rate for all of the target. And the second one is fragment-based drug design and structure-based drug design. This is the key business for our subsidiary Vial in UK. And the third one is that it's a target protein degradation, as I just mentioned that. So this DEL is very suitable for the protein target protein degradation project, either for discovery of POI binders or E3 binders. And the last one is that in the last 2-3 years, we have already set up our synthetic therapeutic oligonucleotide platform. So based on that, so we have also set up our other capability, including the chemistry, protein, bioscience capability, data and information systems. All of these capabilities can provide services outside. And in the past years, so we have been focused on the DEL technology development. And in the last 2 to 3 years, we have published more than 20 different publications focused on the DEL technology development and optimization, including the chemistry development and screening method optimization. And since the founding of the HitGen in 2012, we have already have more than 300 existing partners, and these are the customers in public domain. You can see that, so there are some of the big pharmas like Pfizer, J&J, MSD, Sanofi, Takeda, etc., and also in some of the biotech company like Forma Therapeutics, Sparkle, and Caliber, etc. And yeah, this is just my final slide. So yeah, so welcome to all of the audience to visit us in China, Chengdu, and also in Venalis in UK. Yeah, thank you so much for the listening. Yeah. Olga?
Yeah. Thank you very much. Thank you very much for such a great talk. Me personally, I really big fan of OpenDEL, of this product, because it's such a great idea, to make such a complicated technology like available and accessible literally for everyone. I mean, just yeah, having like a small of the budget, having a target, you can perform a DEL screen and have the results. Yeah, and, so we really enjoy working with you guys, and we will be promoting OpenDEL product for our customers for sure. And if you don't mind, like a few questions from my side. You mentioned that this technology is suitable for products as well, and my question is, do you still need to discover like each part of the product separately and then combine all these three parts together?
Yeah, that is, we are very quite flexible. So some of the customers want to order the DEL kit together, for example 30 DEL together, right? And also before that, after we sign the CDA, so we can also disclose all of the structures to the customers. Customers have the option to select any OpenDEL from the collections. So we are quite flexible. And also in some cases, for example, in the previous collaboration with our other customers, they first order the whole kits, including 30 DELs. After screening, they found that some of the DELs could be very productive against their own specific target. Then they will order this focus library from the DEL, OpenDEL collection. That's quite flexible. That really depends on the customer's request and options.
Yeah, thank you very much. Yeah, another question I have about quality control. How you can check whether your DEL libraries have a good quality or not? So could you please elaborate a little bit more on this?
Yeah, sure. Yeah, this is a very good question. So yeah, before the library construction, so we have a lot of the quality control. The first one is that we call it the chemistry quality control, right? Because that in the library construction there are thousands of the different building blocks will be used in the library construction. So before that, so we have to make sure all of the chemistry used in the library construction are DNA compatible, right? So then first, so we will develop the DNA compatible chemistry to see whether the yields are acceptable, and also the first thing is that whether this chemical reaction is compatible with the DNA, no damage, right, to the DNA species. So this is the first thing. And the second thing is that after the chemistry is optimized, so we will use the optimized chemical reaction conditions to validate all of the building blocks that will be used in the library construction. So then we will set up a criteria, for example, 50%, 60% of the yields. Based on these chemical yields, so we finally selected the building blocks used for the library constructions. So this is the chemistry. So before the library construction. So during the library construction, because in the first cycle it's not post stage, it's just the single component reaction, right? So each chemical reaction is still can be applied using the LC mass to monitor whether these chemical reactions is good or not. After the pooling, so we still have the method. One is to use the high resolution mass approach to see, for example, to add one of the building blocks into the chemical reactions. Either although it's a poor state, we still can see the mass shift before the chemical reactions and after the reaction to make sure this building block can be added to the chemical and to the DNA. This is just and also in 2021 we collaborated with Janssen team to develop a method for the pooling stage quality control for the chemical reactions, and that is published in bioconjugate chemistry. For the DNA tag, it's quite simple because we use 100% of assembly using the GI image to monitor all of the DNA will be added to the DNA. The last thing is that we also use next generation sequencing for all the library to see whether we can see we can find all of the DNA codons as expected with designs. Yeah.
Yeah, thank you very much. It's very helpful actually. And last question from me, it's about whether OpenDEL is suitable for RNA targets.
Sorry, which target?
Yeah, the last question from my side. Is OpenDEL suitable for RNA targets?
RNA target, right? Oh yeah. So in the last year, we have a publication in the nucleic acids, research. So if you have time, so you can. That is an open access paper. So we published a paper talking about using the DNA-encoded chemical library for RNA target selection. But that for the RNA target selection is much more complicated than the traditional target selection because so this is some of the unspecific binding between the DNA and the RNAs, right? So we developed some of the methodology for the target selections and also for the data analysis. So first, so we will add for the RNA target selection. So we will add, for example, the RNA patches, to the selection system to block the unspecific binding between RNA and the DNA species. So this is the optimization during the RNA target selection. The second one is that during the data analysis, so we developed a algorithm to double check whether those signals are from unspecific binding between DNA species and RNA or the specific small molecule with the DNA. So that we have both kind of the methodology. One is data selections, and the other one is the data analysis. You can check. You can see the details in the paper.
Yeah, I mean, using these, let's say, technologies, we can use OpenDEL. We can take RNA target. I mean, we can use these technologies you mentioned in your article and your paper. So, and we can actually do this DEL screen with OpenDEL, or we need some specific library.
So in theory, it's possible, but probably we don't suggest that because without the experience, it's hard to do the RNA target selections. Yeah. Mm-hmm. Okay, that's my suggestion because that is a little bit complicated than the traditional one because as I just mentioned that we will offer a DEL selection menu to our users, but that menu is a much more about the traditional DEL selections. So there is any complicated one, for example, the RNA target selection. So that's have to depends on the expertise from the customers, whether they are familiar with the RNA target and also some of the details on the selections.
Yeah, I see. Thank you very much. Yeah, another question. How do you determine if the molecule is enriched?
Yeah. So so let you can still see my slide, right?
Yeah.
Okay, so I just try to get a. Yeah, we take this one as an example. So on the right side of the plot is this one on the left side is this is a 3-cycle library, right? The cycle 1 is 4 different types of the building blocks. Cycle 2 is a bifunctional group like the aldehyde acid. The third one, cycle 3 is a primary amine or secondary amine. So you can see for this cube there are 3 different plots, right? So R1, R2, and R3. So we will, during the data analysis, we will put, all of the reads, a sequence into this plot. So you can see that. So if the compound was enriched after the screening, so usually there is a feature here. We normally is a, for example, in this case, it's a line feature, right? The line feature means there are 2 cycles are enriched. There is only 1 cycle is variable, right? Take this one as an example. The cycle 1, this is R1. R1 is variable, but R2, R3 are fixed. So that means for this target, probably the R2 and R3 can be very important for the target. So that is a clear sign for the probably the structure activity relationship, something like that. So then we will determine, this is a different bubble here is a different compound. So then after that, we will determine, which compound we will do the off-DNA synthesis to finally determine the activity. Yeah. So probably this is the much more clear here.
Yeah. Yeah, thank you very much.
Sorry. So yeah, so in most cases, it's not the singleton. It's a, in most cases, probably the line feature or plane feature.
Mm-hmm. Yeah, okay, thank you very much. Another question. Yeah, there's a question about pricing, actually. If you could comment on the cost of running a screening against a target, that would be great. Just to get some ballpark estimate regarding the cost. Do you work with small group companies or academic labs?
Yeah, this is a very good question. So currently for our customers, so we previously we did do some of the analysis of the distribution of the customers. Yeah, so some of them are from the academic groups, and some of them from the biotech company, and even for the big pharma. So so I'm pretty sure our OpenDEL product is quite affordable. So even for the academic group can still afford the prices.
Yeah, it's a good question. Yeah, I believe we can let's say discuss this budget and cost estimates over emails. I will yeah, I will share with you like all the questions, all the list of attendees, and you will be able to comment on this. And
sure, definitely, yeah, definitely. So if you have these, because the price is kind of the confidential, so we can communicate through email. Sorry about that.
Yeah, no problem at all. Okay, and another question on slide 16. What is the difference between DEL hit like series and new hit series that shows such high difference in conformational rate?
Okay, so yeah, this is a kind of the AI machine learning stuff. So after the after the selection, so we have a lot of the a vast number of the data after their selections. So you can see that after screening, so we have already identified the active hits. So there are 3 series of compound, right? The activity ranging from 1 micromolar to 4 micromolar. And also we can see that, so all of these compounds, 13 compounds have enriched building blocks, this kind of cyanide group. So then we use this post-selection data for the as a starting point for AI machine learning. So then we to predict the compounds commercially available from vendors. So after that, we found 2 series of compounds. So one series of compound is containing the same moiety as DEL kits, right? So this kind of the cyanide subgroup here. So for this one, for this series, so the AI machine learning, the success rate is quite high. So there are 82% of the success rate to get the active compounds. But so some of the medicinal chemistry is not quite happy with them. Why these compounds are contain the same moiety, right? So then we put effort to see whether we can use the AI machine learning stuff to see it's possible to find new compound without this enriched moiety. So this we call it new series. So you can see here is no cyanide moiety, it's just the hydrocycles here. So for this one, definitely the success rate is lower. We ordered 62% of the commercial available compounds from vendors. There's only 15 compounds active. The success rate is 24%. This is the thing I have to point out here.
Yeah, okay, thank you very much. It's clear now. And probably the last question before we close our webinar, the question is, the DEL data is noisy. How do you post-process the screening results to identify the hit compounds?
Yeah, so let's show the the slides in here. Yeah, here is just one of the very normal selection condition setup. For example, so we are interested in this target, right? There's the ApoNA50 plus, the cofactor acetyl-CoA, right? So this is the add the the main target and cofactor together, and then if we want to see whether there's some of the whether these signals are real or not, so we have to set up the control experiment. For example, the without target, right? Because the target is immobilized on beads. So in that case, the without the target, we can clearly see whether the DEL molecules bind to the beads or bind to the target. So during the analysis, we have to remove the signals from the controls. So we only see the signals only enriched onto the target, not onto the beads or other controlled targets. So that is one of the benefit or advantage of the DEL selections over high throughput screening, because we can set up multiple different selection conditions to compare the signals, to see whether these signals are real or not.
Mm-hmm. Okay, I see. And just to specify, to make it more clear for me, yeah, you show these 3D plots, just like on this picture. So what exactly is pictured on these 3D plots? I mean, I can see, of course, it's error 1, error 2, error 3, but what exactly is
yeah, the R1 is this red color, right? This is scaffold. R2 is here is a different acids. R3 is also the Sec3 is a 1,800 different acid here. The plot all of the NGS data here to see whether there's some of the feature, as I just mentioned, that line feature or plane feature, to see whether the signal good or not.
Okay, I mean the size of this dots.
Oh, the size is relative to the reads of the sequence. Yeah.
Okay.
Right.
Okay. So I believe we're done with the questions. Thank you very much for really nice talk, really interesting talk. It was a pleasure to host you today. Yeah, I hope to to meet sometime in person. Yeah, and thank you very much.
Yeah.
The recording will be available on YouTube, and I will follow up in the email about the link.
Yeah.
Okay.
Thank you so much. Yeah.